we discuss about the third step of the aerobic respiration. This third step of the aerobic respiration is termed as the terminal oxidation. Terminal oxidation or electron transfer system. Or ETS. Very simply, now discuss about this terminal oxidation or the electron transfer system. What is the terminal oxidation? The oxidation of reduced coenzymes into their oxidized, or we can say the original forms, is called as the terminal oxidation. The oxidation of reduced coenzymes, we know during the glycolysis, link reaction, also in the Krebs cycle, respiratory substrates may get oxidized. This oxidation takes place with the removal of the hydrogen. Removal of hydrogen. These hydrogens are accepted by the NAD, and NAD may get converted into the NADH2. Means this coenzyme NAD is reduced here into the NADH2. So again form the NADH2. Regeneration or synthesis of the NAD by removing the hydrogen from these coenzymes, or maybe the sometime FAD, is called as the oxidation. And so to regenerate the coenzymes again is called as the terminal oxidation. Means the oxidation of reduced coenzymes into their oxidized form is related with the regeneration of the NAD and FAD like coenzymes. So this process involves the electron transfer. This process is related with the electron transfer. During the electron transfer, there are so many electron carriers are present. These electron carriers transfer such energy, and therefore this is termed as the electron transfer system or a respiratory chain. And at last, pre-molecular oxygen accepts this electron. At last, the role of oxygen is at the end of this step. Means oxygen is final acceptor. Till this, there are the different type of the electron carriers are present there. These electron carriers transfer the electron from one substrate to the next. And therefore, this is also called as the terminal oxidation. Here, the ATP formation takes place during this electron transfer. Electron transfer occurs. During this electron transfer, energy is released. This energy released is accepted by ADP and inorganic phosphate, and they convert into the ATP. So the synthesis of ATP molecule during the electron transfer with the help of energy released is called as the oxidative phosphorylation. Means this terminal oxidation or the electron transfer system involves so many processes. Oxygen is the last electron acceptor, means the oxidation occurs at the last, therefore it is termed as the terminal oxidation. Energy is transferred by the electrons. There are so many electron acceptors are present, so this is called as the electron transfer system. Also, the synthesis of ATP takes place with the help of energy released during the electron transfer. Therefore, it is also called as the oxidative phosphorylation. And so, how this oxidation of the reduced coenzymes means we discussed earlier, when oxidation takes place, removal of the hydrogen occurs. This removed hydrogen accepted by the NAD, they convert into the NADH2. Now, in the terminal oxidation or the electron transfer system, we discuss about how this reduced NADH2 and sometime FADH2 converted back into their oxidized forms. How? So, when from the NADH2 hydrogen is released, this released hydrogen is accepted by the oxygen. Means hydrogen and oxygen gets combined with each other. And when hydrogen and oxygen gets combined with each other, they forms the water molecule. This formed water molecule, in this process, also necessary in the reactions which are carried out in the Krebs cycle. So, the electron transfer system is a very important process, is somewhat important event in the process of the aerobic respiration. And therefore, here we discuss more about the electron transfer system with the help of schematic representation of this cycle. So, in the terminal oxidation, the reduced coenzymes may get regenerated for the recycling of this total respiration process. How this process occurs, we discuss here in briefly. The electron transfer system here includes the FMN when this process occurs inside the mitochondria. The inner membrane of mitochondria contains the cristae. On this cristae there are the elementary particles or F1 particles are present. These F1 particles have the different type of the electron carriers. Means the process of terminal oxidation or the electron transfer system occurs on the F1 particles of the cristae. Means the NADH2 which get reduced into the glycolysis, link reaction or the Krebs cycle may transfer towards these F1 particles, and then NADH2 may get oxidized to its oxidized form NAD. During this, hydrogen is released. This hydrogen is accepted by the FMN and FMN gets converted into the FMNH2. When FM1 get reduced into the FMNH2, it again gets oxidized by releasing the hydrogen. This hydrogen is accepted by the coenzyme Q. Means here the coenzyme Q gets reduced. Coenzyme Q. Again this coenzyme Q releases the hydrogen, and here the coenzyme Q again regenerated. When this hydrogen is released, this hydrogen may get split into the two proton molecules and two electron. Means here the not only regeneration of the NAD but also the hydrogen which get released from the NADH2 possesses the energy, and hydrogen with this energy moves towards the FMN, towards the coenzyme Q, and towards the various electron acceptors. So this process here takes place in a stepwise manner. Now these electron acceptors are the iron containing substances. Total electron acceptors. These are the iron containing substances called as the cytochromes. So when these cytochromes take the electrons, they may get reduced means this iron containing substance ferric get convert into the ferrous. When electrons given means electrons get oxidized, then this electron iron containing substances ferrous get reoxidized to the ferric. Means when here the hydrogen is released and come towards the coenzyme Q, this hydrogen splits into the protons and electrons. These electrons move from one electron acceptor towards the next electron acceptor. So, these electron acceptors are the iron containing substances generally called as the cytochromes. When these cytochromes or iron containing substances takes the electrons, then these ferric compounds may get reduced to the ferrous, and when electrons are given to the next cytochrome, then again this ferrous may reoxidized into the ferric. And therefore, here we can say that these electron carrying substances or the cytochromes continuously oscillate between the ferric and the ferrous, continuously oscillate, and therefore these electrons move from the first cytochrome B, cytochrome C1 complex. Then electrons move towards the cytochrome C. These are the electron acceptors: cytochrome B, C1, cytochrome C, cytochrome A, cytochrome A3. All these are the iron containing substances. And then finally electron move from the cytochrome A3 towards the oxygen. As here the two hydrogens are released, two hydrogen molecules means the protons are formed, two electrons are moved from cytochrome BC1 complex to the cytochrome C towards the cytochrome A, cytochrome A to cytochrome A3, and then from cytochrome A3 to the finally electrons are accepted by the O, that is the atmospheric oxygen. And so this atmospheric oxygen converts into the negatively charged ions. Atmospheric oxygen means electrons are accepted at the end of this process by the atmospheric oxygen, means the final acceptor is the oxygen. In the aerobic respiration, when we discuss from the breakdown of the glucose molecule up to the formation of the acetyl coenzyme A, or when also in the Krebs cycle, there is no role of the oxygen. Also in the terminal oxidation at the start of this process, or when the electrons are moved, there are no role of the oxygen. Oxygen here comes at the last step, that the electrons are accepted finally by the atmospheric oxygen. And so this is called as the terminal oxidation. Here the terminal oxidation of the electrons may be the takes place. And when this terminal oxidation takes place, meanwhile this protons comes towards the oxygen, and these two protons and this negatively charged atmospheric oxygen may get combined with each other and forms the water. This water formed in this process is called as the metabolic water. And the last, with the help of these protons, here the water molecules are formed. This water is called as the metabolic water. So during this step, electrons are moved from one electron acceptor to the another electron acceptor. Then also the question in our mind is arise, where the ATP are formed? Yes. When electrons or when hydrogen split, energy is released at every place. Yes, energy is released at every place. But not at the every places, but at some places, this energy is utilized in the formation of the ATP molecules. At first, here, when NADH2 is reduced, oxidized, into the NAD and hydrogen is released, here some energy is utilized by the ADP and inorganic phosphate in the formation of the ATP molecule. Means here is the one ATP molecule is formed. Then when electron move from cytochrome BC1 complex to the cytochrome C, also at this step electron releases the energy. This energy is utilized in the formation of the ATP. Here the second ATP is formed. Also, when electron move from the cytochrome C towards the cytochrome A, energy is released, but this energy is not utilized for the formation of the ATP. But when electrons move from the cytochrome A to the cytochrome A3, here also the energy is released, and this energy is released, utilized for the formation of the ATP. Means when a single NADH2 may get split at that time, or NADH2 get oxidized into the oxidized form of NAD, or regeneration of the NAD takes place. Here three ATP molecules are formed. One, when the NADH2 oxidized and hydrogen goes to the FMN, when electrons move from the cytochrome B C1 complex to the cytochrome C, when electrons move from the cytochrome A to the cytochrome A3, here we say that the three ATP molecules are formed. In the similar manner, when the FADH2 is used for this process, FADH2 enters from the coenzyme Q. And so, when FADH2 is present here, FMN does not have the important role. So, FADH2 releases the hydrogen oxidized into its original form FAD, and this hydrogen goes towards the coenzyme Q. This coenzyme Q releases the hydrogen and splits into the two protons and two electrons, and then again come into its original form, that the coenzyme Q. Means when FADH2 is utilized for this process, that the electron transfer system, only two ATP molecules are formed. Means in the electron transfer system, here we can say that from one NADH2, three ATP molecules are formed, and from one FADH2, two molecules are formed. Means here we say the now how ATP are formed, how food molecule gets completely oxidized and more amount of the energy is released to carry out the various cellular activities. मग आता आपल्याला इथे लक्षात येईल की नेमकं इलेक्ट्रॉन ट्रान्सपोर्ट सिस्टीमचं महत्त्व काय आहे. इलेक्ट्रॉन ट्रान्सपोर्ट सिस्टमचं महत्त्व असं आहे मित्रांनो की ही इलेक्ट्रॉन ट्रान्सपोर्ट सिस्टममध्ये NADH2 चं ऑक्सिडेशन होतं, तो त्याच्या मूळ फॉर्ममध्ये येतो आणि त्याच सोबत त्या हायड्रोजनमध्ये जी एनर्जी आहे त्या एनर्जीच्या साहाय्यानं एका NADH2 पासून 3 ATP तयार झालेले आपल्याला पाहायला मिळतात. त्याचबरोबर जर FADH2 असेल तर एका FADH2 पासून 2 ATP मॉलिक्यूल्स जे आहेत हे 2 ATP मॉलिक्यूल्स ह्या इथे आपल्याला तयार झालेले पाहायला मिळतात. म्हणजे आता आपल्या डोक्यात ट्यूब पेटली असेल की अरे खरंच ग्लायकोलिसिसमध्ये 2च ATP तयार झाले होते, क्रेब्स सायकलमध्ये पण 2च ATP तयार होत होते, म्हणजे 34 ATP कुठून तयार होणार आहेत. तर आता 34 ATP ह्या इथे इलेक्ट्रॉन ट्रान्सपोर्ट सिस्टमच्या सहाय्याने तयार होणार आहेत. मग आता आपल्याला एकदा रिमाइंड करावं लागेल की नेमकं हे जे काही समजा NADH2 आहेत हे NADH2 कुठं कुठं तयार झालेले आहेत, कोणकोणत्या प्रोसेसमध्ये किती NADH2 तयार झालेले आहेत हे आपल्याला ह्या इथे लक्षात घ्यावं लागेल. आता आपण ग्लायकोलिसिसकडे वळूया. ग्लायकोलिसिस ही जी काही प्रोसेस आहे ह्या ग्लायकोलिसिस प्रोसेसमध्ये निश्चित आपल्याला सांगता येईल काही NADH2 तयार झालेले आहेत का. आठवा थोडंसं तर निश्चित आपल्याला सांगता येईल की ग्लुकोज मॉलिक्यूल स्प्लिट होत असताना 2 पायरुवेट सोबत 2 ATP आणि 2 NADH2 तयार करतो. मग आपल्याला माहित आहे एका NADH2 पासून 3 ATP तयार होतात, म्हणजे ग्लायकोलिसिस मध्ये 2 NADH2 आणि 3 ATP मॉलिक्यूल्स तयार होतील. असं जर गृहीत धरलं तर हे 6 ATP ह्या NADH2 पासून तयार होतील. त्यानंतर लिंक रिॲक्शन, लिंक रिॲक्शन मध्ये काही ATP तयार होत आहेत का, तर लिंक रिॲक्शन मध्ये ATP तयार होत नाहीत. पण लिंक रिॲक्शन मध्ये ऑक्सिडेटिव्ह डिकार्बोक्सिलेशन घडतंय. ऑक्सिडेटिव्ह डिकार्बोक्सिलेशन घडतंय याचा अर्थ ऑक्सिडेशन घडतंय, याचा अर्थ तिथं पण एका पायरुवेटचं रूपांतर एका ॲसिटिल कोएन्झाइम A मध्ये होत असताना एक NADH2 म्हणजे 2 NADH2 इथं तयार होतात. आणि मग हे 2 NADH2 एका NADH2 पासून 3 ATP, 2 NADH2 पासून 6 ATP ह्या इथे आपल्याला तयार झालेले पाहायला मिळतील. त्यानंतर आता आपण क्रेब्स सायकलकडे वळूया. क्रेब्स सायकलमध्ये ज्यावेळेस चार्ज ऑक्सिडेशन होत आहे, हे चार्ज ऑक्सिडेशन झाल्याच्या नंतर 3 वेळेस 3 NADH2 आणि 2 ज्यावेळेस क्रेब्स सायकलच्या टर्न पूर्ण होतील त्यावेळेस एकूण 6 NADH2 तयार होतील. 6 NADH2 एका ATP NADH2 पासून 3 ATP, 6 NADH2 पासून 18 ATP तयार होतील. आणि ज्यावेळेस दुसरा आपल्याला सांगता येईल की ज्या ऑक्सिडेशन घडलेलं आहे त्यावेळेस त्या तिथे आपल्याला सांगता येईल 2 FADH2 तयार झालेले आहेत. हे 2 FADH2 2 ATP तयार करतील, म्हणजे 4 ATP ह्या इथं तयार होतील. मग आपण हे सर्व ATP जर का इलेक्ट्रॉन ट्रान्सपोर्ट सिस्टममध्ये मोजले तर 18 आणि 4 22, 22 आणि 6 28 आणि 6 34 असे 34 ATP जे आहेत हे 34 ATP ह्या इथे आपल्याला तयार झालेले पाहायला मिळतील. आणि निश्चितच ग्लायकोलिसिसमध्ये 2 ATP तयार झालेले होते आणि क्रेब्स सायकलमध्ये 2 ATP तयार झालेले होते, म्हणजे 34 आणि डायरेक्ट तयार झालेले 4 ATP असे मिळून 38 ATP large amount of the energy or total energy which is present in the complex food material may be released or break down in the terminal oxidation, and so from one glucose molecule when completely oxidized here the 38 ATP molecules are formed. आणि अशा पद्धतीने मग या इथे आपल्याला सांगता येतील की 38 ATP जे आहेत हे 38 ATP तयार होतील. म्हणजे वेगवेगळ्या पद्धतीने ज्यावेळेस इथं आपण विचार करतो त्यावेळेस आपल्याला आता क्लिअर कन्सेप्ट सगळी झाली असेल की आपण ही जी रिस्पायरेशन आहे, ह्या रिस्पायरेशनच्या व्याख्यामध्ये डेफिनेशनमध्ये असं म्हटलं होतं की एनर्जी रिलीज होत असताना स्टेप वाईज रिलीज होणार आहे. ही एनर्जी स्टेप वाईज रिलीज होते, एकदाच रिलीज होत नाही. एकदाच रिलीज जर झाली एनर्जी तर सेल बस्ट होण्याची शक्यता नाकारता येत नाही. आणि म्हणून आपल्याला इथे सांगता येईल की एनर्जी ही टप्प्याटप्प्यानं वेगवेगळ्या जागेवर एक एक ATP तयार होतो. इलेक्ट्रॉन ट्रान्सफर सिस्टममध्ये सुद्धा एक एक NADH2 मॉलिक्यूल वापरला जातो. आणि हा एक एक NADH2 मॉलिक्यूल ज्यावेळेस वापरला जातो त्यावेळेस तो सुद्धा स्टेप वाईज 3 ATP तयार करतो. आणि म्हणून आपल्याला इथे सांगता येईल की पूर्ण रिस्पायरेशनमध्ये 38 ATP तयार होत असताना ATP कुठेही डायरेक्ट तयार होत नाहीत. ATP हे अतिशय सुंदर रीतीनं टप्प्याटप्प्यानं तयार होतात. त्यामुळे आपल्या शरीरामध्ये एकदाच हीट किंवा एकदाच एनर्जी तयार न होता स्लोली एनर्जी तयार होते. आणि ती एनर्जी आपल्याला वेगवेगळ्या पद्धतीनं वेगवेगळ्या टप्प्यावर वेगवेगळ्या सेल्युलर ॲक्टिव्हिटीज पार पाडण्यासाठी मदत करते. जर ही सगळीच एनर्जी एकदाच शरीरामध्ये तयार झाली तर आपलं शरीरामधल्या ज्या सेल्स आहेत ह्या सेल्स डॅमेज होण्याची शक्यता असते. आणि म्हणून त्या दृष्टिकोनातून ह्या इथं स्टेप वाईज एनर्जी रिलीज होते. हे रिस्पायरेशनचं फार महत्त्वाचं वैशिष्ट्य आहे असं आपल्याला सांगता येईल. आणि मग ह्या 3 स्टेपमध्ये ह्या इलेक्ट्रॉन ट्रान्सफर सिस्टमचं सिग्निफिकन्स आता तुमच्या लक्षात आलं असेल की द इलेक्ट्रॉन ट्रान्सफर सिस्टीम ऑर द टर्मिनल ऑक्सिडेशन जनरेट्स मेजर अमाऊंट्स ऑफ द एनर्जी इन फॉर्म ऑफ द ATP. हा टर्मिनल ऑक्सिडेशनचं महत्त्व काय आहे तर सगळ्यात जास्त ATPसाठी जी काही एनर्जी आहे ही ATPची एनर्जी ह्या इलेक्ट्रॉन ट्रान्सफर सिस्टममध्ये रिलीज होते. 34 मॉलिक्युल्स out of the total 38 molecules are produced through the ETS. 38 ATP मॉलिक्यूल जे काही एका ग्लुकोज मॉलिक्यूल पासून तयार होणार आहेत त्यापैकी 34 ATP मॉलिक्यूल हे ह्या इलेक्ट्रॉन ट्रान्सफर सिस्टमद्वारे तयार होणार आहेत. त्याचबरोबर इट रिजनरेट्स ऑक्सिडाइज्ड कोएन्झाइम्स सच ॲज द NAD and FAD from their reduced forms NADH2 and FADH2 for recycling. जे काही NADH2 आणि FADH2 तयार झालेले होते ग्लायकोलिसिसमध्ये असतील, लिंक रिॲक्शनमध्ये असतील, क्रेब सायकलमध्ये असतील, ते रिसायकलिंगसाठी पुन्हा वापरता येण्यासाठी सुद्धा त्या NADH2 आणि FADचं रूपांतर NAD आणि FAD मध्ये होतं. म्हणजे प्रत्येक वेळेस NAD FAD तयार करण्यासाठी सेलला वेळ वायाला घालवायची किंवा त्याची एनर्जी वायाला घालवायची गरज नाही. ह्याच पदार्थांचं रिसायकलिंग झाल्यामुळे सेलवरचा जो लोड आहे तो लोड कमी होण्यास मदत होते. इट ऑल्सो प्रोव्हाइड्स वॉटर मॉलिक्युल्स नेसेसरी फॉर द क्रेब सायकल. वी see in the electron transfer system when the atmospheric oxygen combines with the electron, this oxidized electron may get combines with the proton molecule and forms the biological water. This biological water is used in the process of the क्रेब सायकल. क्रेब सायकलमध्ये घडून येणाऱ्या ज्या रिॲक्शन्स आहेत त्या क्रेब सायकलमध्ये घडून येणाऱ्या रिॲक्शनसाठी सुद्धा अशा प्रकारचं हे जे काही बायोलॉजिकल वॉटर आहे हे तिथे वापरलं जातं. इट रिलीजेस एनर्जी इन अ स्टेप वाईज मॅनर टू प्रिव्हेंट द डॅमेज ऑफ सेल. मी म्हटल्याप्रमाणे जर ही सगळी एनर्जी एकदाच रिलीज झाली तर ती एनर्जी आपल्या शरीरात फायदेशीर ठरवण्याच्या ऐवजी ती आपल्याला घातक ठरेल. सेल डॅमेज होतील, सेल डॅमेज आहे तर टिश्यू डॅमेज होतील आणि मग आपल्या शरीराला वेगवेगळ्या त्रास वेगवेगळ्या समस्यांना सामोरं जावं लागेल. आणि म्हणून एनर्जी ही स्टेप वाईज मॅनरमध्ये रिलीज होणं गरजेचं आहे. अशा पद्धतीनं हे जे एरोबिक रिस्पायरेशन आहे ह्या एरोबिक रिस्पायरेशनचं जे मेकॅनिझम आहे ह्या एरोबिक रिस्पायरेशनच्या मेकॅनिझममध्ये ह्या 3 अत्यंत महत्त्वाच्या स्टेप आहेत. Glycolysis is the common pathway between the aerobic respiration and anaerobic respiration. But Krebs cycle and electron transfer system are occur only into the higher type of the living organism in which the double membrane bounded organelle like the mitochondria is present. So these steps are the how aerobic respiration occurs we discuss in this and earlier sessions. In next session we discuss about the anaerobic respiration, how the microorganisms like the bacteria are also in some time in our human muscle cells. Such type of the anaerobic respiration occurs. How this process occurs we discuss in the next session. Okay, thank you.